Hello! This video is made as part of the final project for the Bioprocessing Technologies and Downstream Processing course. In this video, we will talk briefly about the production method for fungal silanase. First of all, what is fungal silanase? Silanase is a hydrolytic enzyme that can hydrolyze silane into simple sugar such as xylose. This enzyme has many applications. They can be used in the baking industry, paper industry, animal feed supplements, as well as pharmaceutical products. This enzyme can be produced by microorganisms, one of which is fungi. Fungal silanase is essentially silanase that is produced by fungi. The most commonly used fungi are filamentous fungi such as the Aspergillus genus. Some of the examples are Aspergillus niger and Aspergillus awamori. There are essentially three steps for producing fungal silanase: fermentation, extraction and purification, and lastly formulation. The fermentation process is also called the upstream part of the production process, whereas extraction, purification, and formulation are all part of the downstream process in the production process. Speaking of the fermentation process, there are two types of fermentation or rather fermentation reactors that will be covered here. The first one is submerged fermentation, where the fungi is submerged in liquid media. The second type is called solid-state fermentation, where the fungi is grown on solid media as a support. Both of these fermentation types have many parameters which can be optimized to maximize fungal silanase production. The first is the substrate. Pure sugar such as glucose is often used in submerged fermentation, but due to the high cost of using pure substrate, agro-industrial waste can be used as an alternative substrate. Agro-industrial waste is often used in solid-state fermentation, but they can also be used in submerged fermentation. Some of these include wheat straw, corn cob, sugarcane bagasse, as well as palm oil empty fruit bunch. Aside from substrate, temperature as well as aeration are also important factors to be considered. There are also factors that are more specific to the type of the fermentation. For submerged fermentation, there are agitation using baffler as well as pH control throughout the fermentation process. Solid-state fermentation also has an exclusive factor, which is the initial moisture content for the fermentation medium. The next aspect of upstream production is the control system of the fermenter. Solid-state fermenters typically use feedback control, whereas the submerged fermenter typically use a combination of feedback and feed-forward control. The last factor in the upstream process is the cleaning process. After fermentation has been done, the fermenter needs to be disinfected and sterilized periodically. The most common way is by cleaning in place or CIP system, which is made of 5 steps. The first step is pre-rinsing using reverse osmosis or potable water. The next step is alkaline circulation using sodium hydroxide, followed by a round of rinsing. Then acid circulation is performed using organic acid, followed by the last round of rinsing. The fermentant that can be prepped for the next batch of operations. Following fermentation, the next step is extraction and purification as well as formulation. These steps are the downstream part. This part can be quite flexible. Using only extraction methods usually only produce lower purity silanase, whereas using a combination of extraction and purification can produce silanase with higher purity. Both of these silanase have their own field of usage. The first aspect of downstream processing that we will cover is the extraction process, which essentially recovers the product from the fermentation mix or media. There are two general types. Filter paper such as the Whatman filter paper is commonly used to extract silanase from submerged fermenters. The second method is buffer or solvent extraction, which is usually used to extract silanase from the solid support of the solid-state fermentation. The next step in the downstream processing to produce an enzyme with higher purity is purification. There are 4 general methods for this. The first is ultrafiltration, which involves using membrane filters. The second is precipitation, where precipitating agents such as ammonium sulfate, ethanol, as well as polyelectrolyte are used to precipitate the silanase. The third is chromatography. There are 2 types of chromatography generally used for silanase purification, which are ion exchange and gel filtration. The last method is aqueous two-phase system or ATPS, which involves a mixture of polymer-polymer or polymer salt dissolved in an aqueous solution, which will form 2 layers. The silanase can be retrieved from one of the 2 layers. Regardless whether purification is performed after extraction or not, the last step of the fungal silanase production is the formulation or the packaging step. Fungal silanase can be packaged in 2 different stages, liquid and powder. For liquid, the product from extraction or purification can simply be placed in a container, mixed with stabilizer, and then stored in an appropriate temperature, in which storing in 4 degrees Celsius can make the enzyme last longer compared to room temperature. For packaging in powder form, there are 2 options. One is spray-drying, which uses high temperature to evaporate the liquid, leaving only the product in powder form. The other option is enzyme immobilization, which have an advantage of producing reusable enzyme. That is all of the information we could provide in this video. Hopefully the information was useful. Thank you so much for watching. Beyond this point is the credit, which includes the source of the pictures.